Welcome, and thank you for joining the ASPPH Presents webinar, Innovative Screening Methods for COVID-19. I am Dewana Huff, the CDC Health Department liaison assigned to the state of California.
I'm Jennifer Byrne. I'm CDC's Task Force Liaison Officer to the Vaccine Task Force.
Jennifer and I work on the COVID-19 emergency response team with the CDC, and we are pleased to co-moderate this session today. COVID-19 has changed the world we live in. In order to return to some level of normalcy, we need people to be vaccinated, and we need to have effective screening methods. We are excited to hear from 4 groups today who have developed some of these innovative screening methods and have utilized them effectively in the educational setting. From using animals as screeners to COVID test vending machines, we'll discuss how these methods have come about and how they might be applied to additional settings. As we hear from the speakers, after we hear from the speakers, we will have time for questions and for you to make comments.
Now it's our honor to introduce your speakers for the day. Dr. Rebecca Fielding Miller is an assistant professor at the University of California, San Diego in the Herbert Wertheim School of Public Health and the Division of Infectious Disease and Global Public Health. Nikos Gerfield is the county veterinarian and head of the Vector Disease and Diagnostic Laboratory and the outreach group in the San Diego County Vector Control Program, Department of Environmental Health. Dr. Jesse Jokhurst is an associate professor in the Department of Nanoengineering at UC San Diego. Dr. Jokhurst completed a PhD in chemistry at the University of Texas and completed postdoctorate work at Stanford Radiology. Lisa Joyner serves as the Director of Health Promotion Services at UC San Diego Health, where she oversees wellness education for UC San Diego students. Laura Cohn graduated from the University of Washington with a master's degree in public administration. She is a leader in the field of education, early childhood, and workforce development. Patricia Unger founded the Kensington Veterinary Hospital in San Diego in 1991. She is the founder and president of Sense Solution, a startup business focused on using dogs to help resolve public health challenges. Now we'd like to invite Nikos and Patty to begin.
Great, thanks for inviting us to speak. As you know, dog scent detection abilities have been used for many years for hunting, for tracking, finding drugs and explosives, and even for detecting some medical conditions like different cancers. Next slide. Last April, early in the COVID pandemic, the University of Pennsylvania put out a news story that they had trained dogs to accurately detect samples from patients with COVID-19. This was an intriguing thought because any tool that could be used to combat COVID-19 could be useful. Next slide. So canine olfactory anatomy, physiology, cell biology, and even genetics are like a superpower. Their sense of smell is 100,000 times more sensitive than ours in detecting volatile organic compounds at a sensitivity of 1 part per trillion. That's like smelling 1 teaspoon of sugar dissolved into 1 million gallons of water. Some of the factors that give them this amazing ability is a nasal airflow, where 12% of inspired air is directed to an olfactory sinus for the purpose of olfaction, rather than to the lungs for gas exchange. Air is sampled during both inspiration as well as expiration, which is the function of those slits at the sides of dogs' nares. The dog's olfactory bulb is 40 times larger than humans relative to the rest of the size of the brain. It has 50 times more olfactory receptors with many more sensory cilia per cell than humans do. And they have 3 times as many olfactory genes as people. These are just some of the many evolutionary adaptations of the canine nose and olfactory system. Next slide. So recognizing the potential for canines to contribute to the fight against COVID, there's been a handful of studies that have tested this idea. Next slide. A study from Germany published last year tested dogs' ability to detect salivary and tracheobronchial secretions from 7 hospitalized symptomatic COVID-positive patients compared to 7 non-infected patients. 8 dogs were trained for 1 week and then run through over 1,000 randomized double-blind trials where they performed with an average of 83% sensitivity and 96% specificity. Next slide. A recent bioRxiv paper by Grandjean et al. Used sweat samples from 151 symptomatic COVID positive and 110 COVID negative hospitalized patients that were matched for various conditions. Using dogs from a variety of working backgrounds, after over 1,300 trials, the dogs achieved between 71 to 100% detection accuracy. Next slide. So what are the challenges of taking these preliminary studies to the next level? While they were promising, additional rigorous testing is needed to assess the limit of detection, the sensitivity and specificity over the course of COVID disease. Can dogs detect asymptomatic as well as symptomatic infected individuals and distinguish between other conditions? There are regulatory obstacles as well. Dogs are not covered under current FDA regulations, and the actual volatile organic compounds that they detect are not known. Future validation studies must be done to assess sensitivity and limit of detection in comparison with authorized antigen and PCR tests, and their ability to distinguish infection from potentially confounding factors like concurrent diseases, different ethnic backgrounds, skin microbiomes, and other factors that could potentially affect VOC signatures. Lastly, do new SARS-CoV-2 variants or vaccine status result in different VOC profiles that must be considered? Next slide. So with that, I'd like to turn it over to my co-presenter, Dr. Patty Younger, who will describe some examples of how COVID detection dogs are currently being used. Thank you.
All right. Thank you, Nikos. Thank you so much for asking me to speak today. I'm really honored to be included in this group. I find it really exciting that there are so many interesting, useful, and novel approaches regarding management of COVID. The past year has had more than its share of challenges. It's also opened up many opportunities, what I refer to as the silver linings. And many of these are in the area of care, diagnostics, and management. Hang on, my video's not on. There we go, sorry about that. Okay. I'm excited to spend a few minutes talking about the COVID sniffing dogs. Here we go. When I first saw the announcement from University of Pennsylvania last year that they had successfully trained a few dogs to sniff COVID with high accuracy, I found this really exciting. I presumed we would see more such projects and rapidly given the dog's low-tech efficiency and accuracy, but surprisingly, that's taken longer than expected. As a practicing small animal veterinarian for the last 36 years, I already appreciate how incredibly talented dogs are. To see this special olfactory talent being put to use for the betterment of public health makes total sense to me. Next slide, please. Thanks. In July, the UAE announced that they were employing trained COVID sniffing dogs to screen incoming passengers at their airports, using a swab of a sweat sample from the passenger. Those with positive alerts are then sent for confirmatory on-site lab testing. Dubai is, of course, one of the busiest airports in the world. They trained 21 dogs, age range 1 to 6 years old, 1 female, the rest are males. I don't know why there are so few females. 17 of the dogs had been previously trained to detect explosives. 2 were cadaver search dogs. 2 had no previous experience. There were 8 Malinois, 5 Shepherds, a mix of German and Dutch Shepherds, 6 Cocker Spaniels, 1 Labrador, and 1 Pointer. Across the board, they averaged about 92% accuracy in repeat tests, and their accuracy did not seem to be affected by previous scent training. Next slide. Shortly after that, Helsinki, Finland began using COVID sniffing dogs to screen incoming passengers at the airport there. They also used the sweat samples, reported similar accuracy to the UAE. Currently, Australia, France, Germany, Chile, Russia, Iran, and Britain also have dogs in training. But overall, the use of these dogs has been limited by the lack of political will and funding, even though the use of these dogs is far more economical and faster than conventional testing. It only takes a dog about 8 to 10 seconds to screen a person. And as Dr. Gerfeld mentioned, some of the early studies, they were using saliva and tracheobronchial secretions. That's obviously not practical in a real life application. But these recent studies are showing that using the sweat samples works very well. Next slide. Earlier this year, you may have heard that the Miami Heat announced they'd be using dogs to screen all incoming fans to their games, starting in March this month. This will allow them to safely accommodate more fans in their arena and help get their community back to more normal function faster. To my knowledge, this is one of the first commercial applications of COVID sniffing dogs. These dogs are also being used to screen classroom and public spaces at Florida International University, where some of these dogs were actually trained. Next slide. So how are these dogs actually trained? It's actually surprisingly simple. As Dr. Gerfeld describes so eloquently, COVID, like many other diseases, produces a unique volatile organic compound scent profile, which appropriate dogs can easily be taught to identify. It's done in a very positive way. The dogs learn the odor and they're rewarded with a treat or a ball or a new toy, something that they want. As he describes so thoroughly, their olfactory system is extremely well developed, which has obviously helped them survive throughout the millennia. Many breeds can excel at this. Pretty much any working dog with a drive to work, especially those that are scent motivated, can be trained to do this. In the initial studies, they only needed a week to train the dogs, but it appears that using 2 to 3 weeks of training produces better results, allows the dogs a little more time off so they can recharge. You get a happier working dog. Our current plan is to start with 2 purpose-bred dogs, likely dogs that have either been bred to sniff explosives or possibly cadaver, probably not narcotics, and 2 to 3 shelter or rescue dogs. Next slide. Many are probably familiar with Ian McKay's model of the Swiss cheese approach to COVID control. The concept here is that given the complexities of COVID, it's unlikely that any one single modality, testing, track and trace, quarantine, vaccination, etc., will solve the problem. It will take a combination of many levels of Swiss cheese with holes that do not fully align to get us back to something resembling normal. COVID sniffing dogs provide one more welcome modality, just another slice of the cheese. Next slide. I truly believe these amazing dogs can provide another great resource to help get our community functioning at a more normal level and faster. In my opinion, the previous studies that were done were really not validated rigorously enough against other real life applications. They were measured primarily against how well did the dogs track on the samples, not on people, and also looking at their effectiveness against variants and other confounding factors like other diseases, and more recently, the impact of vaccination. These are challenges that we're planning to address in the design of this study, because I think before we can put these dogs to work, we need to know that those are reliable and accurate. Thank you so much for your attention, and everybody keep up the great work out there. Thank you.
Awesome. Thank you for that, Dr. Patty and Dr. Nikos. Yes, I share the sentiments of Christy Peterson, who said, fascinating, another reason to love dogs. So I noticed that we have some questions coming in for the chat. I'm going to ask everyone that if you would like to ask a question, we encourage you to do that early in the presentations by putting that in your Q&A at the bottom of your screen. There is a Q&A button at the bottom of your screen. If you will put your questions there, so when we have time at the end, we will go ahead and answer those questions for you. And then we also encourage you to, well, we will also be recording. So we want to let you know that we will be recording and we will make this presentation available shortly after the session is done today. Thank you. I turn it back over to you, Jennifer.
Thank you. Now we'll move on to our next panelists, Rebecca Fielding Miller and Laura Cohn.
Hi there. Thank you so much. I'm Rebecca Fielding Miller. I am faculty at the University of California, San Diego, and I will be presenting on a program that Laura and I have really been thinking about and working to develop since, June or July. called the Safer at School Early Alert Program. And this is really a collaboration between UCSD and the County of San Diego. So there's a lot of conversation about how we get kids back in school safely. And we didn't accidentally mean to stumble into the sort of biggest political hot button this year, but here we accidentally are. But I think that one thing that everybody can agree on is there are definitely some kids who need to be in some schools with some adults, right? We know that the communities that are being hit hardest by COVID-19 are simultaneously the communities where kids are experiencing the highest level of learning loss, the less access to technology support, enrichment support, and who have parents who have the most need to be outside the home to work, parents who are most likely to be essential workers who can't work from home. And so what we wanted to do at UCSD is take some of the amazing technology that we've developed for our campus, and Lisa Joyner is going to speak about that shortly, but think about how can we use technology that was initially developed to keep our campus community safe and really use it for communities across San Diego County, where kids are kind of being hit hardest by both the pandemic and the effects of school closed. And we thought we want to take the sort of best technology, the sort of cutting edge, and create really a preferential option for the most vulnerable communities. We think that this is something that is necessary for low-income communities, communities with a lot of immigrants and refugees. And so just to give you a sense of where we're working in 10, 12, actually 13 right now schools, 10 elementary schools and 3 childcare sites all around the county of San Diego. And what I want you to see from this map is that dark blue is places with a lower health equity score, and green are communities that are doing a bit better. And if you look at our map, you'll see that all of our dots, all of our partner schools are in areas that are dark blue or light blue. And for those of you who don't know, San Diego is very close to Mexico, and we're working in several schools right along the border as well. And this is sort of where the current epidemic is in San Diego. These are data as of yesterday. So you can see, just like everybody else, we had our really big spike in December and January, and numbers have really come down. They've started to level a little bit now as the shelter in place has been lifted. And that's this graph on the right. On the left, you can see the number of cases per 100,000 stratified by age. And you can see, just like everywhere else, kids under 10 really do appear to have the lowest incidence, rapidly followed by people in their 70s. And it's been kind of cool to watch that number decline as more people get vaccinated. And then we have folks in their 20s and 40s, sort of people who are working, who are a bit higher. And I think one thing that's important to keep in mind about this is we are looking at this, we're not seeing this youngest age group start to go up yet in San Diego County, but it's definitely something we have our eye on as the new UK variant comes out based on studies we've seen from Israel and Italy. So what are we doing? We have developed this thing called SASE, the Safer at School Early Alert Project. And SASE is a combination of environmental monitoring and responsive testing for elementary schools and childcare. So there's a few different components of it. A major piece is wastewater detection. So they're literally called robots, which I think is quite fun. These sort of 2.5 feet tall, 2 foot in diameter little cylinders that sit on top of like a sewage clean out and have a little tube that goes into the sewer and just kind of slurp up sewage every 15 minutes. And we have that because, as I think a lot of you know, we can actually, sewage is actually a great way to monitor for COVID-19. In fact, you might actually be able to detect COVID-19 little pieces in feces even before somebody would test positive on a PCR on a nasal diagnostic swab. The other thing that we have as part of SASE is surface monitoring. So teachers basically take a fancy Q-tip, the same swab basically that you would use in a diagnostic test, and at the end of the day, they rub it on the floor. And for all of our partner schools, every day we send those jars of wastewater and those diagnostic swabs off to a lab at UCSD. And in about 36-hour turnaround time, we can say, we think, even though everybody on campus is asymptomatic by definition because you're doing symptom screening, we think somebody either on campus or in this particular classroom has COVID-19 or is shedding the virus. And so it's necessary to do responsive testing, to go and do diagnostic testing so that we can identify who this case is and support them to isolate safely and support their cohort to quarantine safely. The other thing we can do too is, in the meantime, until we've identified who that person is, we can say, somebody in this room, somebody in this school is shedding virus. This is a really good time to reinforce prevention measures, to really make sure that your airflow is good, that you are double masking if necessary. Maybe think about spending some more time outside. This is a great week for bonus recess. So you can see it's a complicated system. And one thing that we think is quite cool about it, and also a little bit overwhelming, is when we think about translational research, this sort of basic science to preclinical, the clinical, all the way up to public health. We're basically doing all of this at once, which is both very exciting and very overwhelming. So we are sort of simultaneously refining surface swab techniques to monitor for settled pieces of COVID on the floor, which nobody thought we could do before we tried it out in some school settings. We're simultaneously refining that. We're simultaneously figuring out, can you actually monitor school sewage flow for COVID? Because as several people pointed out to us when we began this project, not everybody poops at school. And what does it look like to implement this project at scale and in a way that would potentially be sustainable and affordable in the long term? That's a lot. So just to go into a bit more detail, the components of SASE are environmental monitoring, looking for people who are asymptomatic but infected through wastewater monitoring and surface sampling. Once once we think there is a case on campus, we then come and provide on-campus response testing. And we've discovered that that on-campus piece is actually quite important because it removes a lot of barriers to testing access. Nobody has to go make an appointment. Nobody has to go find who will actually order a COVID test for a kid, because it turns out some pediatricians will still not order COVID tests for kids. And it's quick. And then we also know that it's really important that all of the communication, all of the conversations about, here's why we're asking to test your kid at school, here is what this wastewater signal means, here's why we're asking you to mask a little bit more thoroughly this week, that all of these health communications are done in a way that are sensitive to cultural and economic and community context. And like I said, we've very purposefully done this with partner schools who serve immigrant communities, who serve refugee communities. San Diego actually has one of the biggest refugee resettlement communities per capita in the country, and communities that are experiencing a lot of poverty and other issues. Because if we can design something that works for communities at the margins, then you can design something that works for everybody. But when you design something for communities that don't have a lot of access issues, then you just have something for communities that don't have access issues. So there's a bit of work to this process, but basically, these are our robots. And you basically need to install this robot, usually a sewer clean out or manhole cover. The kids like to decorate it. This one on the left is Harry Botter, which is my favorite. You can see they actually named it, it's Harry Botter and the Rancid Water is the name of this one. The greatest student from Botwarts. And this one on the right is named Georgie. It's also been a great STEM opportunity. The kids have had lots of great conversations with the scientists. And then the surface swabbing every day, which is fairly straightforward. So we've been doing a week of PCR testing for validation, and now we're moving on to response testing. We did 12 weeks of PCR testing for validation, and now we're moving on to response testing. And I just, before I run out of time, want to give you a quick sense of how the system works. So you can see that getting people to consent to testing is not small. But after about 12 weeks of engagement, this bit here on the right, you can see that the number of external diagnostic tests really dropped, while the number of tests done with our SASE program really went up. And that was a testament to our team and the trust that they built. This is just another kind of cool example. And what I want you to see up here on the left is that we had a bunch of yellow under surfaces, which is to say we had a bunch of rooms that looked like maybe there was something on campus. We had some wastewater that seemed like it was positive. And then over the weekend, a staff member tested, it turned out they were positive, and then everything went clear the next week. So the answer of can we detect asymptomatic cases seems to definitely be a yes. I'm going to skip over this for the meantime and just add that making sure that this is also grounded in social and behavioral concepts to think about people as people and making sure we meet people where they are is crucial. Otherwise, it's just a robot. It's not public health. I'll stop there. I have so much more to say about SASE, but I know my time is up.
Great, thank you. Are we ready with the slides for our next speaker? Great, all right. Dr. Jockhurst, when you're ready.
Great, well, thanks for the introduction and thanks for organizing. My name is Jesse Jockhurst. I'm on the engineering faculty at UCSD. And today I'm happy to talk to you about some work we're doing in colorimetric sensing for COVID-19. So the audience is probably familiar with PCR testing, and this is by far the most common testing that is being done in our society that involves a swab and then some amplification. What's great about PCR is the ability to amplify basically single copies of the virus. And so the ability to do this kind of wastewater detection that my colleagues were describing would not be possible without this really amazing amplification that PCR can do. It's also incredibly precise, right? The ability to discriminate between different viruses. The challenge lies in this need for kind of a centralized lab. And so there's also a lot of work in portable PCR. Maybe people put them into things that become increasingly smaller, but there's some fundamental need to heat and cool the sample that really kind of limit the ability to do this at point of care. There's also this kind of lag time. And so you'll probably remember at the beginning of the pandemic, people would wait days and days to get the results back. And that's certainly become better. It's well below 24 hours at UC San Diego now, but it's still not instant. And so, in this, when people are starting to interact with more people, this becomes a real concern. So I think we've talked about the advantages, the disadvantages, and still kind of the expense of PCR. One alternative is serology, and so this is what people kind of refer to as the antibody test, which tells you if you have been have you been exposed or not. And so what's nice about these kinds of approaches is that they can be quickly kind of customized into something similar to the home pregnancy test, where one line versus two lines, and that report out whether you have been exposed to the COVID virus. It doesn't necessarily tell you the current status, right? Because once you have the antibodies, at least we think you should have the antibodies for life. So the difficulty here is it's less cumbersome because this kind of can be put onto the footprint of a home pregnancy test. It's shorter, but we just don't have the sensitivity and the specificity and the real-time insight that we need. And so what myself and many other labs are working on is the concept of surveillance. And I know that's part of the theme of today. And I like using this analogy of a smoke detector. And a smoke detector is constantly doing surveillance in your home, right? And most of us don't spend much time thinking about our smoke detector, right? Except for when it goes off. And when your smoke detector goes off, it doesn't necessarily tell you it's a grease fire versus an electrical fire, or it doesn't say there's a fire in the bedroom versus the kitchen. It just says there's a fire and you need to take action. And so as we move into the next stages of the pandemic, as cases hopefully become more and more rare, surveillance may become a more attractive method to complement testing. And so that's one area that my lab is working on, is thinking about ways that we can do surveillance of COVID-19. So the marker that we're using is these not relatively complicated proteases. A protease, if you're not familiar, is just a protein that eats another protein. And so the protease that we're monitoring and using for surveillance is called MPRO, which is short for the main protease. MPRO is produced in cells as part of the viral life cycle, and it allows the virus to replicate once it infects the host cells. And so there's many therapies that are looking at inactivating MPRO. But what we wanted to do is think about, could we use MPRO as a diagnostic, in contrast to the nucleic acid that is used in PCR or the antibodies that are used in serology testing. So we can collect MPRO from a variety of sources, and we're still, digging down in detail about what are the exact concentrations in these different kinds of samples. It's definitely present in nasopharyngeal swabs, and as well as saliva and exhaled breath condensate, although the concentrations between these do change dramatically. And we're still working on getting higher numbers of samples to really understand this distribution. The other thing that we're inspired by is what are we using for surveillance? And so at this point in the pandemic, there's really nothing that's more ubiquitous than masks, particularly in high density settings that are particularly vulnerable to outbreaks. Masks are still a really important frontline part of the Swiss cheese model. And what's great about them is that they're also inherently collecting saliva, exhaled breath over the course of the day. And so this is, I think, a really beautiful electron microscope image of what a N95 respirator looks like. And so you can appreciate these different layers, and you can also imagine that as you're breathing on this or maybe through this, depending on the type of mask that you have, that this is kind of an ideal setting to use this as a tool to collect some of these samples and then potentially use that for surveillance. And so that's what we're working on. When we first started thinking about this, the concept of, well, do you integrate this sensor into the mask, or is it something that goes on top of the mask? And I think the right decision was to have it be a kind of a sticker device that goes on top of the mask, so that it's not wed to any one type of mask. And so what we're building is this sticker that you would put either on the inside or the outside of your mask and breathe on it over the course of the day. And if any of this main protease accumulates, at the end of the day, when you squeeze this little blister pack, this liquid will flow through this chamber. One of the little lanes has a positive control, so this will automatically change colors. The other lane has these vents that you would be breathing through, and so this would be the test lane. And as this liquid moves through the channels, a color change from pink to purple indicates that there is indeed MPRO present. So what is this liquid? This is kind of the key to this technology, and we're basically using either dyes or nanoparticles. I'm showing nanoparticles here. And what's interesting about gold is that when you think about, everybody knows what gold looks like, but as you make it smaller and smaller and smaller, it starts to have a color that changes based on its size. And so if you have very small gold particles, they look red. And if you have bigger gold particles, they look purple. But if you put a bunch of small particles together, it starts to act like a big gold particle. So here at the bottom, you have a bunch of small particles that act like a big particle, and the color changes. And this is something you can really clearly see by eye. And so by using this protease and some cleverly designed peptides, we can dictate whether these particles remain as individual monodispersed particles or they aggregate and have a color change. And so you can see this really obvious kind of color change in some of these samples in the laboratory, where we either expose the particle to the protease or not. And then we're looking at the sensitivity in a variety of different samples. And so we're exhaled breath condensate still works quite well. Saliva has many other proteases. Saliva is just a tough sample to work with. And so one of the things we're working now is comparing exhaled breath condensate to saliva, and then also comparing this to some of the commercially available reagents. So there's the liquid part that has been a big challenge. I feel pretty confident about that. There's also a major engineering element in actually building this blister. So the nice news is it's pretty inexpensive. I mean, our cost of goods right now is 50 cents to make this, and you would assume it would go down as these things scale. And they're basically made out of vinyl, cotton, and these absorbent pads that are well established in the field. So again, this is not going to be sitting there broadcasting someone's COVID status. You would simply wear it like this, and at the end of the day, you break this blister pack and the fluid flows and you observe for a color change. And so, you know, if you wanted to do that in front of people, you could. If you wanted to do that in the privacy of, you know, your home, your car, somewhere that was protected, you could do that as well. So this is, there's no activity until you break that blister pack. So this is kind of a prototype. And so here you can see it before this blister pack has been squeezed. And afterwards, you're seeing this obvious color change in the lane that has the M-Pro protease versus the control lane. And so this happens really over the course of about 5 minutes. It's instantaneous. It's single use. You would throw this away after single use and then put on another one for another day. So the next steps are kind of further refining just the ability to optimize the sticker construction to make enough for it to be usable, answering basic questions about just where does the sticker go on the mask? How does it change as a function of different kinds of masks? How long does a positive person need to wear one of these before they break the blister pack? And so to do that, we're validating this in positive patients, and then comparing this to PCR in larger cohorts. So we really only had funding on this for about 6 weeks, but I'm pretty optimistic about the progress that we've made, that we can start using these in positive patients in the second quarter. So I just, while I've got an audience, I always mention that recruiting talented students to Southern California, and particularly there's a lot of exciting work that I think could be done between the School of Public Health at UC San Diego and the Jacobs School of Engineering. So thanks again for the opportunity.
Wonderful. Thank you, Dr. Joe Kirst. Now we'll move on to our final panelist, Lisa Joyner. Are you ready?
I am ready. I think you all have the slides. Perfect. So thank you all for having me. My name is Lisa Joyner. I'm the Director for Health Promotion Services here at UC San Diego Health. And I want to talk to you a little bit about one of our innovation projects that we worked on last year through a program called Return to Learn. Last spring quarter, we really figured out that students would be moving home. And we had some students who would also be staying here with us on campus. And so a group of us got together, known as Return to Learn, and we started to work with Natasha Martin, who is one of our scientists on campus who has really heavily been studying COVID-19. And she did some modeling, predictive modeling, to figure out how could we safely bring students back here in the fall quarter, with knowing that UC San Diego has around 38,000 students who are here with us on a very frequent basis. And so what we wanted to do is start a pilot. And that pilot consisted of testing around, or offering testing to around 10,000 students, but also our students who were off campus living in San Diego County, or those who were coming from nearby cities like Los Angeles. We also wanted to test our campus employees, and we wanted to potentially get around 1,500 to 2,000 tests a day. This is a very lofty goal with, at the time we were talking about mid-turbinate, where individuals were saying, oh no, I have to stick a swab all the way up to my brain. But we really wanted to figure out an efficient means of testing for our student population in our campus community. And so after meeting with individuals from Operational Strategic Initiatives, our Student Health and Wellbeing Cluster, and then also our Jacobs Hospital, we decided that our goal would be self-administered testing for our students here at UC San Diego, and we would start that with our first pilot. Next slide. Our pilot was really going to be centered around engaging students in testing, education and awareness, and letting them know about opportunities to test here on campus where they live, learn, and play. And so what this is right here is our first part of it will be, we had it in around 8 residential communities that consisted of grad and family housing, that also consisted of our undergraduate colleges. We wanted to bring testing where the students lived. So that was really us creating access to testing. We created detailed instructions, and multiple iterations is really highlighted here because I probably created over 25 sets of those instructions as we were learning more and more about the testing process, what worked well and what did not work. We actually partnered with our public health department on campus, Dr. Nancy Binkin, and she was able to email out to our student population to learn more about what was preventing them from testing and then what was also encouraging them to test. The big part of the encouragement piece was people wanted to keep their family and friends and loved ones safe. The negative part of that piece would be that students were afraid of the testing. They were afraid to use the swab and they didn't want to go too far. And with our self-administered testing, we had clinical staff on site, but it was provider observed, meaning that an individual was standing there, they were watching to make sure that you went deep enough to collect the sample. With this, we really demonstrated feasibility because we had around 1,500 students who had self-tested. They were going daily. They wanted to know their COVID-19 tests, whether it was positive, whether it was negative. Individuals at this time wanted to travel home, and they felt like getting a test would be very important. So in this phase, we really used the mid-turbinate. We also made sure that we were updating our instructions for our students. And we also used an app, which we'll talk about in our next couple of slides. So next slide, perfect. So this right here is some of our instructions that we use. This is probably midway through where we had really start to refine what it was that we were doing. These instructions were to help them. So a student would walk up to this table, they would go ahead and use the hand sanitizer. They would also take out their phone. Or we had an iPad nearby if they did not have a phone because they really needed to download the UC San Diego app. This app tied their tests back to the individual. We still had our clinical staff on site, and we were continuously updating the instructions, which leads us to where we are now, which we'll talk about a little bit later. Everyone was still spread 6 feet apart or more because we wanted to ensure safety. And these desks and tables that they were surrounded by were wiped down on a frequent basis. Next slide. So this is our self-identification sample. Right here, what you notice to the right is our media or our vial, and there's a barcode on here. So as students would come up to get their kit, they'll take out their phone and they'll scan that barcode. What you see to the left is a picture of the UC San Diego app, and you'll see it says sign in to scan your COVID-19 test. Whether you're a faculty, staff, or student here at UC San Diego, you have the ability to do this. This makes sure that this sample is tied back to you. So they'll scan that barcode on their testing vial every time that they do a test here at UCSD. Next slide. So what we started to move to, and we talked a little bit over last summer about, do we stick with mid-turbinate? Do we go to saliva? Saliva at the time had not been validated. And so we knew that students were still afraid to use the long swab and go to the mid-turbinate. So we moved to anterior nares. Anterior nares just really covers that bottom portion of your nose. And we gave students instructions that they would be able to utilize. Undergraduate move-in for us consisted of around 9,000 students, and they were tested a part of the move-in process. So as students were coming up, we had identified a parking structure or parking lot that they could do a drive-through testing site. And so we implemented one of our first drive-through testing sites for our students where it was provider tested. So individuals would go through the lot, we would self-screen them, looking at different types of symptoms that they may have in regards to COVID-19. And our staff members from Student Health and Wellbeing and also our hospital would go ahead and do a test for them. So this was on site, it was clinical, they had their family members in the car, but this helped us really to get students in a space of learning how to test because they would be doing testing weekly as they live back on campus. What you see to the right is one of our testing sites called our Price Center B testing site. This site is still operational over at our student center, and it sees faculty, staff, students, and health science employees. And so this site was also implemented in September of 2020, along with our drive-in site for P102. Next slide. So we came upon another space of we needed to create access to testing. Actually, I was coming back from visiting my family and I saw a vending machine in the San Diego airport. And here we are today with our COVID-19 vending machines. We have since deployed over 11 machines and each machine holds over 600 kits. So in a day, our vending process does around 2,000 tests for our students. And to the left of this machine is actually a bin where an individual would drop back their kit. I want to go to the next slide and I want to tell you a little bit about how this works. So what you see here is a kit that you would get out of the machine. We deployed 11 machines. We also have one machine in athletics because many folks in athletics have to test more than once a week. So they're typically testing around 2 to 3 times depending on competition. The kit contains your media, your swab, and your instructions. And at the table where individuals are getting their kits right next to the vending machine, we have our peer-to-peer educators or our Triton Health ambassadors. They are there to support our students to answer any questions that they may have, or if there's trouble at the vending machine. Next slide. So these are our instructions for our students. We have them in Mandarin, we have them in Spanish. They're also online. Really details out what they need to do. And because of all of the iterations that we had with our instructions at the very beginning of our Return to Learn program, we have now really created a solid instruction plan for individuals who are taking our COVID-19 tests. Next slide. So this is our current vending machine. The vending machine has a hand sanitizing station, a drop box next to it, and we also pick up over 2 times a day, and this is open 7 days out of the week. Our lab turnaround time for results are around 16 to 17 hours, and results go right back to the student's My Student chart or the faculty or staff members' Epic chart and My chart. Next slide. So currently, these are all of our locations of vending machines on campus. We are expanding to 20 locations, and we're also putting in validation. Validation will ensure that individuals are not creating or taking more tests than are needed. So they'll have 2 per day that they're able to take. Right now, because of the vending machines, and typically you'll see a vending machine for candy or soda or something that you want to buy, there is a $1 charge that is immediately removed. So tests are free to our students. It's free of charge for faculty, staff, and students here at UC San Diego. We're still doing over 2,000 kits or tests per day. We have created access and we continue to do that. Students can use the vending machines. They can go over to our Price Center locations, or they can use some of our health system sites as well, which are drive-through. As we're continuing to expand, we will also have 2 testing locations that will be drive up at our parking decks here at UC San Diego. We will continue to look at ways in which we can create access to testing for our campus community. I believe that's it. So thank you all very much. I appreciate it.
Thank you, Lisa, and thanks to everyone, all of the panelists for sharing with us today. So we have some audience questions, and actually some of them have already been answered. The panelists have graciously been answering some of the questions in between the presentations. So make sure that you look in the Q&A and see the answered questions as well. And as a reminder, you can enter your question by clicking on the Q&A button on the bottom of your screen. So we're going to take this remaining time to answer as many questions as possible. I think Dewan is going to start us out.
Awesome, awesome. Thank you, Jennifer. So our first question that hasn't been answered yet, could the mass sensor pick up someone who is positive sitting next to or or in the same room as the person wearing the mask?
That was our hope. And then if you think about a smoke detector, it's detecting the whole room. And so we were hoping that this would be able to offer broad surveillance of everyone that's around the sensor. I don't know if it's going to be able to do that, as we have started to do some preliminary testing. I just don't know that the detection limits are going to be high enough. So there will still be a lot of value if each sticker is unique to each wearer. There's a different kind of information that will offer, similar to true testing. And so, yeah, but the short answer is that we just don't know yet.
Great, thank you. I'm actually looking at one question that came in the chat box, and this is for all panelists. Will approximate cost of each of the testing methods be discussed during this presentation? So if you could speak a little bit to the costs of the different methods that you've all presented on.
So I can speak to the costs of SASE a little bit. So a piece of doing this has been obviously thinking about how much it's going to cost, because that's a really relevant question. We have also been working with Natasha Martin, because I don't think she sleeps. I think she just does nothing but model and cost things out day and night. So the little robots cost about $2,500, $3,000 each. The surface swabs are pretty inexpensive. They're about the same, honestly, as a diagnostic swab, and we think that you could probably use a lot of the same technology that you would use for a diagnostic. So a lot of the cost in this environmental monitoring is really in the personnel and in the process of getting the samples from the school site to the lab, and then sort of running the samples for $30 to $40, $50 each, just really depending on what lab you're working in. So it depends is the short answer, but it was really designed to be something that was both more cost efficient than weekly diagnostic testing, which is just going to be a really time and resource intensive process, and something that would catch people who might not be interested in asymptomatic testing. So to get more people with less money.
And I see someone asked a little bit about the cost of the vending machines. We get that a lot. Currently, we have 30 vending machines here at UCSD with around 20 being deployed. And I believe I spent around $125,000 on those machines. The great thing, though, is that, you know, with student fees and students not being here, the big question is that coming out of our student fee pool? It is not. The University of California Office of the Chancellor and the President, they have decided to incur that cost. And so that has been what's been really good about that, is we're able to offer the testing at no cost to students. Now, if some community wants to come and test here at UC San Diego Health, they can do that for a very minimal cost of $65. And I say minimal because you can go out in the community and spend a couple $100 on that as well. And so we are trying our best to keep costs very low for our students, because everyone is in a different situation, and create access for our students as well.
And I can answer regarding for the dogs as well. The main application for the dogs is not on an individual basis, but more for event basis. Let's say if you've got a large group of people who are going into a Padres game or into a concert or event, it could be hundreds to thousands of people. The dog cost is going to work out at something looking like about $1,000 a day. There's a breakdown though. It's going to be variable depending on what they're doing, how long they're working, how many dogs. But in all the models so far, on a per person basis compared to individual testing, the dogs are meant as a screening, not as a testing. It's quite a bit less expensive. So what makes them efficient for group events.
Yeah, and I mentioned that we're about 50 cents a piece, but all of these surveillance approaches kind of have to be cost effective, or it's just not going to work.
All right. So the next question we have is for you, Lisa, or anyone that may know. Do you see vending machines being available for high school campuses?
We do. We have actually talked to quite a bit of schools, high school, universities, in helping them to implement vending at their institution or their campus. I think it would be a big collaborative effort, though, to make sure that they're collaborating with a lab that can run those tests, because here at UCSD, we have our own labs. And so that's where the results are being run on campus, whether it's in the COM lab or whether it's in one of our other labs on campus. So it is possible. It's funding, and it's going to make sure that you're tied back to some type of hospital and EMR and medical record system as well.
Thank you.
Okay, our next question. So this question comes from a student and a student worker at Arizona State University that works for the COVID-19 testing sites on campus. Says, we implemented the saliva-based test that I enjoy using as a student. Would you consider transitioning to a saliva test to help increase testing, or do you believe the nasal swab is successful for your campus?
Nice, Arizona State, whoever you are. I see you're anonymous. My best friend is the Vice President of Student Affairs at Arizona State. So good question. We have actually considered saliva. It's been a hot topic of conversation recently, where we actually had saliva and our enteronaries testing going at the same time in the Price Center. Why we did not move forward with it is that our campus community, they're already so used to our current testing process. And to make sure that people are on the same page, they understand the testing method, we didn't want to do a big switch. Also, too, what we have found, and we found this through our lab, is that people think they're interested in saliva, but after they sit there and they do it, they actually prefer to just go ahead and do the enteronaries and be done with it, because it's a lot quicker. With saliva, you really have to sit there and gather. You can't eat 30 minutes before. There are a lot of different protocols that you have to make. So we actually have decided not to go with saliva here at UCSD, but we do know some others are doing it.
Thank you. With the dogs, what is the pushback from those who test positive by dogs?
It's an interesting question. The dogs, again, are not testing. So I think let's use Miami Heat as a good answer, because that's the most commercial application. I think the airports are doing the same. The dogs alert on someone saying, you smell like you might have COVID. The airport in Miami Heat then recommends that the person receive, there should be someone on site offering a point of care to a rapid test to confirm it or not confirm it. I think it's new enough that we don't know for sure how much pushback there is. I think by and large, it's been really well accepted at the international airports. Very few, really no reports of people not accepting it well. And that's the model they're using, where the dogs screen, they alert and say, this person smells a little off, they're recommended to get testing. I think what the Heat was saying is, if someone does it, if they're not willing to test, they're just going to say, you can't come in, you have to come back another time. And I think that'll be probably the application in commercial facilities.
Great, thank you. Dawana, do you think we have time for one more, or do we need to close out?
Maybe one more. Let's see.
Okay. See. So if a dog detects COVID, is that individual offered an on-site test to confirm, or are they asked to leave?
Again, that's a decision that's going to be up to the facility, but I think, in my opinion, what should happen is there should be an on-site test offered. If you're using the dogs to screen, then you want to take it through to, if the dog says this person might have COVID, the facility shouldn't be letting that person in unless there's a yes or a no on it. And they would be referred for probably an on-site testing to get that answer on site and get it rapidly.
Thank you.
All right. Well, this has been awesome. So I want to thank all of the panelists for your time, and all of your, and think of all of you for your attention and your thoughtful questions. And again, there will be a recording of this webinar. It'll be available as soon as we're done with it today on the ASPPH website. As you close out your session today, please take a moment to complete a very brief session survey. Zoom will display it on your web browser, so that should pop up for you. And just remember, it's not too late to register for the 2021 ASPPH annual meeting next week. Student rates are available, and you can learn more about these plenary speakers and others as well as register and the links at the bottom on this of the slide. That will conclude today's webinar. We thank you so much for joining. Have a great day.